Dear children, welcome to online classes of biology. So today we are going to start a new chapter, that is chapter 9, biomolecules. So you know the living organisms are made up of what? Chemicals, isn't it? We have studied that the structural and functional unit of life is cell. So what are the cells made up of? Cell organelles. And when we studied the structure of plasma membrane, I told you that the plasma membrane consists of what? Basically lipids are there, proteins are there, isn't it? Phospholipids are there, or proteins are there, and this proteins and lipids, ultimately what it contains, it contain elements like carbon, oxygen, etc., isn't it? So if we do the chemical analysis of organisms, understood, in plant tissues or animal tissues or microbial paste, whatever it is, the elements which we get are what? Finally carbon, hydrogen, oxygen, okay, and other what kind of minerals, etc. Understood? So same thing you will get if you start doing a chemical analysis of earth, okay, or the soil. If you do the same chemicals, you will get, isn't it? So the elements present in earth's crust, okay, and also that is present in the living organisms, they will be same, they are same of their nature, okay. So that these elements you will finally get. How can you conduct, or how can you know that what are the elements present in a living organism? For this, we have to do the what? Chemical analysis. Okay, so chemical analysis, how we'll prepare? We have to take the tissues. Okay, living tissues like vegetable or any animal tissues we have to take, and we have to grind it in trichloroacetic acid. Okay, trichloroacetic acid, what, using a mortar and pestle. Okay, so in a mortar and pestle we have to add the tissues, and then we have to add what? Trichloroacetic acid, and then you have to grind it. Okay, so you will get a slurry, and this slurry, if you filter, okay, through the cloth, a cheesecloth or cotton, will find two fractions. Okay, the fraction, the one, okay, called the one is called the filtrate. Okay, the filtrate, and it is the acid soluble pool. Okay, and the another one is, okay, the retentate, or the acid soluble fraction, sorry, insoluble fraction. So two fractions you will get. One is the acid soluble fraction, and the other is acid insoluble fraction. Understood? So many of the organic compounds are present in the acid soluble pool. Okay, so in the acid soluble portion you will get a number of compounds, okay, and that can be separated. Okay, it can be separated by different techniques, and we can analyze it. Okay, we can purify, and then we can analyze it, and that will give us some idea about what all molecules are there. Understood? So we can even probably know the, we can know the molecular formula, and also the structure of the compound. Okay, so like this, all the carbon compounds that we get from living tissues can be called as the biomolecules. So what are biomolecules? All the carbon compounds that we are getting from a living tissue is called what? Biomolecules. Understood? And what this, in addition to biomolecules, there are some inorganic molecules also, isn't it? So that, how we will come to know what are the inorganic molecules? The inorganic molecules, how we can identify? By doing a destructive experiment. Means you have to take the animal tissue or plant tissue, and then you have to dry it. Okay, so the dry, that is known as what? Dry weight. When all the what the moisture content. Okay, moisture content, if it is getting lost, then that remaining material, okay, is known as the dry weight. So how you will get the dry weight? Okay, you have to take some amount of a living tissue and dry it. Understood? And if you get the weight of the tissue when you are collecting, that what you are getting when you are weighing the animal tissue, okay, that is known as the wet weight. And when you dry it properly, when all the moisture content, okay, evaporates, then what is left is the dry weight. Okay, so wet weight and dry weight is clear. Wet weight means when you collect a tissue and weigh it, it will have the water content or moisture content, that is known as the wet weight. Okay, and when you dry it, okay, and when all the water content is getting evaporated, the remaining material, that is known as the dry weight. Understood? Now you have to burn it. What for you are doing this? To know about the inorganic elements. Okay, so when you burn it, what will happen? Okay, carbon compounds will be oxidized to gaseous forms like carbon dioxide, then water vapor, etc. will be removed. Okay, so after that, what is left out is the ash, isn't it? And ash will give you the idea about what all the inorganic elements are there, like calcium, magnesium, etc. Understood? So, like this, what you can identify the non inorganic molecules also. So, in addition to the what elements which are found in the ash, we can also see sulfate, phosphate, etc. are found in acid soluble fraction also. So, like this, elemental analysis will give you what kind of elements are there in the tissues, isn't it? Like in the form of hydrogen, oxygen, chlorine, carbon, etc. Okay, and compound analysis. Okay, while analysis for compounds, that will give idea of what kind of organic and inorganic constituents are present. Okay, so we can identify different groups. There are tests also, isn't it? You are doing different tests. So for different what functional groups, isn't it? Like aldehyde, ketones, aromatic compounds, etc. We can what identify by different tests. Understood? So like this, we can understand what kind of elements are there in a tissue. Okay, and when we compare these elements with the Earth's crust, the elements which are found are same only. Okay, and when it is found in the carbon and compound form in the living tissues, we call it as what? Inorganic molecules. Okay, so biological body or the body of an organism is made up of what? Various kinds of molecules. Okay, inorganic and organic molecules. Clear? So these functional groups I told, isn't it? They contain different functional groups. So we can study them as what? Amino acids, nucleic acid, nucleotide bases, fatty acids, etc. in biology. Okay, so we'll study as amino acids. So have you heard about amino acids? What is the specialty of amino acids? They are the building blocks of proteins, isn't it? They are what? Building blocks of all the proteins are made up of amino acids only. Understood? So you know different types of proteins. Have you heard about globin? Okay, albumin, keratin. Likewise, different types of proteins are there, and all these proteins are made up of amino acids. And when you see an amino acid, what are the speciality of it? They'll contain an amino group and an acidic group. Okay, understood? So acidic group will be there, amino group will be there as substituents on the same carbon. Okay, and that carbon is known as alpha carbon. Understood? Then so only they are also known as alpha amino acids. Okay, actually they are substituted methane. Methane's formula you knowing now, isn't it? CH4. So they are substituted. Okay, the CH4 hydrogen atoms in the methane is substituted by different compounds like what? amino group over the other amino group or the acidic group. Understood? So like this, here you can see different amino acids like glycine, okay, then alanine, serine, okay. So these are amino acids. So you can see it's containing, or it's a substituted methane, I told, isn't it? Carbon with 4 valencies of hydrogen, okay. Carbon's 4 valencies are filled by hydrogen atoms in what methane. But here you see it is substituted by COH, okay, in one side, and another it is substituted by NH2, okay. So this is the simplest amino acid, which is that glycine, okay. COH, CH2, NH2. Understood? And then alanine, you see in that the one more hydrogen atom is substituted by one CH3 group, okay. And here you see in serine it will be substituted by another CH2OH group, okay. One of the hydrogen of serine is substituted by what? CH2OH. But you can see COH and NH2 will be common. Is it clear? So like this, what you can see that amino acids are what? Only the substituted methane. Understood? Substituted methane. And the speciality is that they will have one or more, okay, amino group and acidic group substitute on the carbon. Understood? So this is the reason why they are known as what? Substituted methanes, okay. There are 4 substituent groups occupying the 4 valency positions. That can be what? Hydrogen, carboxyl group, amino group, and a variable group, okay, that is represented as R group. And based on the R group's nature, amino acids can be of different types, okay. And whatever it is, you can understand that all the proteins are made up of this 20 amino acids. Understood? Only 20 amino acids are there, and with that only all the proteins are made. Just like with 26 letters or alphabets of English, we can make a number of words, isn't it? And words can be used for making sentences. So in the same way, okay, amino acids are only 20, and by their different combinations, there are different types of proteins, okay. And the R group, I told one amino group will be there. What are the different molecules that substitute the carbon valency in a amino acid? This can be hydrogen, carboxyl group, amino group, and a variable group that can be substituted by hydrogen, carboxyl group, okay. Likewise any, okay, amino group and a variable group designated as R group. So like this you can see here one R group is there, and that can be substituted by any of the what molecules, okay. So if it is a hydrogen, I told it's the simplest type of amino acid and is known as what? Glycine. And if it is a methyl group, okay, then we call it as, or that is the alanine. Hydroxymethane, methyl, then it is serine. Understood? So like this, many what amino acids with a common structure. What is the common structure? There will be an alpha carbon, and in that there will be amino group, COH group, and there will be an R group. R group can be substituted by any of the molecules. Understood? Then the chemical and physical properties of amino acids, okay, are due to the what? The groups, that is amino group, carboxyl group, and R functional group. And based on how many carboxyl groups are there and how many amino groups are there, the amino acids are classified into different types, okay. There are acidic amino acid. Acidic amino acid means what? They contain what? COH group more. One COH group will be there in the alpha carbon and in the R, okay. If they are having more than, means more what? Carboxylic group than the amino group present in the amino acids, then it's an example for a what kind of amino acid? Acidic amino acid. Example for acidic amino acid is glutamic acid. Okay, so glutamic acid is also is a amino acid. Okay, what is the speciality? It is acidic, means more acidic group is there, or COH group is more in them. Okay, or it can be basic. Basic means more amino group will be there, isn't it? And example for this is lysine. Lysine is an example for a basic amino acid. Understood? Or it can be neutral when the number of amino groups and what COH group is same. Understood? So there are aromatic amino acids. Okay, aromatic amino acids means what? They are containing aromatic compounds like tyrosine, phenylalanine. Sorry, these are examples for what? Aromatic amino acids, which all? Tyrosine, phenylalanine, tryptophan. So when they are called as aromatic amino acids, when they are having what? The aromatic substitutions. Okay, so this is clear. So if you see an amino acid, okay, it's a substituted methane, I told, isn't it? Substituted methane. Methane will have carbon with 4 what? Hydrogen, isn't it? CH4 is what? Methane. So you can see the hydrogen will be substituted by what? The various molecules like or functional groups like amino group, okay, and the COH group. Understood? And then you can see the other group that is considered as R. Okay, so this is R. There will be an amino group and there will be a what? Acidic group which is attached to the carbon, and then you can see an R group. So R group in alanine, in glycine, you can see it's only having H, hydrogen. Okay, and in alanine you see this R group is having CH2. Okay, and in serine you can see CH2OH. And now in this substitution or in the R group, if the number of acidic group is more, okay, than the amino group, then the amino acid will be a acidic one. And if the number of amino group is more, then it will be a basic. And if the number of amino group and the carboxyl group is same, then it will be a neutral amino acid. Example of acidic amino acid is glutamic acid. Example of basic amino acid is lysine. Neutral amino acids example is valine. Okay, aromatic amino acids examples are tyrosine, phenylalanine, okay, tryptophan, etc. Understood? And now I am going to tell you about another property of amino acid, that is ionizable nature of the amino acid. Okay, that is resulting in the formation of a Switter ion. Okay, Switter ion means what? It carries charge. Okay, means in this amino group they will have positive charge. Okay, how? By adding one hydrogen, okay, atom from carboxyl group to the amino group. Okay, so in solutions it can exist as charged molecule. Understood? So this property is known as what? Or this type of what amino acids are known as what? Switter ion. So amino acids can exist as what? Switter ion. So what are Switter ion? They are having positive and negative charge. Due to this, the structure of amino acids changes in solutions of different pH. Okay, so this form is known as B form is known as what? In this diagram you can see, okay, this is known as what? Switter ion. Means what will happen? The H plus ion from COH will go to amino group, and there amino group will have what? Positive charge, and the carboxylic group will have negative charge. So this form is known as the Switter ionic form. Means molecule carrying what? Positive and negative charge. Understood? So this, if we change the solution, the structure of amino acids also will get changed. Okay, you can see this is one and this is another. Understood? So the middle one is known as the Swifter ionic form. So what we learned today is about the biomolecules. What are biomolecules? The molecules which make up the body of living organisms, okay, or which contains carbon and its compounds, isn't it? Are known as what? Biomolecules. So how will we analyze the biomolecules? By what? The grinding. By grinding the animal or plant tissue in trichloroacetic acid using a mortar and pestle. Okay, then we have to filter it. And when we filter, what will happen? We'll get two fractions: acid soluble fractions and acid insoluble fractions. Understood? Then and in the acid soluble fraction we can find a number of what molecules, isn't it? And by chemical analysis we can identify the substances. So all this, okay, carbon compounds that we get from living tissues are called what? Biomolecules. And in addition to the biomolecules, there are inorganic molecules too. So how we will get the inorganic molecules? By what? By drying it. Okay, then we'll get the dry weight, and then that material we have to burn. So when you burn, what will happen? The water content, okay, will get evaporated, and what is remaining is called the ash, and that contains what? Inorganic elements like calcium, magnesium, etc. Understood? So like this, what if we analyze, okay, the tissues, animal or plant tissues, will get what? They are made up of various elements like oxygen, hydrogen, chlorine, carbon, etc. Okay, and it will be same as that of the what that we get from the soil. Understood?